Hello, my name is Benjamin Filches. I'm a PhD researcher at KU Leuven, and today I will talk about the artificial intelligence algorithm that will be further enhanced within the FOGIT project, a project where we aim to work towards personalized and automated freezing of gait detection. Freezing of gait is defined as the episodic absence of forward progression despite there being some intention to walk. It carries several adverse effects, and the advancement of novel treatments is severely hampered by the difficulty to objectively assess the severity of freezing. The current gold standard lets users perform some standardized protocol, which is then simultaneously recorded by a video camera, and based on this video data, a clinical expert can then manually annotate each freezing segment. However, this is very time-consuming. The objective of the present study is thus, given a standardized trial of some variable length, to automatically recognize and temporarily localize each freezing segment. The present study uses data that is recorded within a motion capture lab, thus the input to the AI algorithm are the 3D optical markers in time. We let users perform a standardized protocol consisting out of straight ahead gait, 180-degree turning, and 360-degree turning. Now I will try to explain the intuition behind the developed algorithm. Prior work in freezing detection mainly used window-based classifiers, where a standardized protocol would first be segmented into shorter length windows of some fixed duration, and each window would then be classified with methods ranging from simple thresholding techniques towards high-level temporal models driven by deep learning. However, window-based classifiers already implicitly impose a trade-off between the model expressivity and the sensitivity to model short episodes. Therefore, we treat freezing detection as a sequence-to-sequence classification problem where deep learning models are used to generate a label for each single input sample. Thus, if we operate at a sample frequency of 100 Hz with motion capture, we generate a label for each single millisecond, thus allowing for very fine-grained freezing segmentation. The present study developed the algorithm that was termed multi-stage graph convolutional neural network. The graph convolutional part of this term relates to the fact that we use graph convolutions that aim to better capture the spatial temporal structure that is inherent to motion capture. Spatial because the optical markers are connected by bones, and temporal because optical markers are connected within consecutive frames. Because we operate at such high temporal resolutions, we observe that a single stage of those graph convolutions is insufficient because over-segmentation problems tend to occur. Over-segmentation meaning that a single freezing episode would instead be labeled as two separate freezing episodes. To tackle this problem, we add multiple stages of temporal convolutions, which aim to enhance the initial predictions, and this results in more enhanced and better freezing segmentation. From the segmentations, we can then compute two quantitative outcomes, namely the percentage time frozen and the number of freezing episodes. The percentage time frozen is defined as the cumulative duration of all freezing episodes divided by the total duration of a standardized trial. And here we observe that the correlation between the model and the manual annotations of a clinical expert are very high. However, for the number of freezing episodes metric, we do observe that the model tends to significantly overestimate the number of episodes. In the future, we would also like to transition from the motion capture lab towards a at-home environment, meaning that we will no longer record movement with a motion capture system that is of course constrained to a motion capture lab, but we will move towards wearable sensors to capture the movement. Patients will also be able to walk more freely rather than doing a fully strict standardized trial. If you would like more details, I can refer you towards the full paper at the following hyperlink, or you are also always free to contact me or one of my co-authors.